Hello, Bio30. Welcome to our first video on genetics on monohybrid or autosomal crosses. We are going to get started talking about a very important man, Gregor Mendel. Yes, Gregor Mendel. Gregor Mendel was a monk, a German monk to be precise, and he was what we would call the founder or the father of modern genetics. He studied pea plants and was really looking at, he was a German-speaking Austrian monk, sorry. He really looked at how do plants pass on their traits. So he established some really key rules for heredity that we still use today. So we call them Mendelian genetics. It's very cool. If you are a fan of Animal Crossing, the game, you do Mendelian genetics if you are trying to breed flowers. And we'll get into that at another time. Mendel focused on something that is called the monohybrid cross, mono meaning one. So we're only talking about one trait. Mendel was a successful geneticist because he ended up developing what we would call the pure lines, and he kept very statistical notes. And I'm going to explain the difference between some key words as we go through this. So why did he choose to study pea plants? Well, he chose to study pea plants because they were super easy to see color, shape, the shape of the pod, the color of the pod, the plant height, where the flowers were, and flower color. So it was very distinct. It was either this or that. You have a chart in your notes that kind of shows you the dominant and recessive traits that he was able to see as he was working with pea plants. So keywords that we need to talk about are pure breeding or true breeding. True breeding or pure breeding just means that they're a homozygous plant. Homozygous means they have the same letter at this, and they're either both capitalized or both lowercase. If they're both capital, it means it's the dominant trait, and if they're lowercase, it means it's the recessive trait. How do you achieve what we would call, in this case, pure breeding or true breeding in plants? It's what we would call selective breeding. You take progeny from a set of parents that show very consistent and predictable traits, and you then breed them, okay? So Mendel would take two tall plants, and he would then have 100% tall offsprings. And anytime he repeated this experiment and repeated this experiment, he always got tall offspring. So what he said was, purebred organisms are also homozygous, which I've already said. So tall plants, breeding with tall plants, give you homozygous traits. So let's set this up in your notes so you can kind of see what we're saying. The trait, let's use the trait and the plant height to study this. So cross one. Cross just means I'm going to mate two things together. So I'm going to take a pure tall plant and a pure short plant. And what happens is when I did that, 100% of the time I got tall plants all the time. So that's what we would call the F1 generation, the first generation. I'm now going to take two F1s. So two plants from the F1 generation. They're all tall. They all look tall. Okay. And I'm going to mate them together. We're now going to have what's called the F2 generation. What ended up happening though is when I did that, I don't end up with 100% tall plants anymore. I now end up with 75% tall plants and 25% short plants. What this reasoned, or what Mendel then said, oh, what this means is the gene for a tall plant is dominant and the gene for a short plant is recessive. Why? Because when a tall and a short were mated together, you only got tall. But their genes were now mixed. They had both a tall allele and a short allele. So when they were mated together, two of those plants mated together, we were able to see a variety of different, what we call phenotypes, or what we see with our eyes. This is an example of a monohybrid cross because we're just looking at one trait. So we're just looking at the height of the plant, nothing else. So the summary of Mendel's cross goes a little something like this. We always are going to use stuff like P1, F1, and F2. P1 means the parental generation. F1 means the first generation. F2 means the second generation, meaning we always are combining the first generations together. And that is kind of the overall legend that we have. So what he ended up doing is it was creating a monohybrid cross. When we do monohybrid crosses, we are always going to set them up the same. And in fact, we are going to set up almost all genetic problems the same using something called a Punnett Square. And I'll show you what that looks like. So whenever you do a problem, we're going to follow these stages as we go through. One, you're going to write down a legend of letters that tell you both dominant and recessive traits. Then you're going to write down the genotypes of the parents. If you are unsure of a genotype of a parent, you will leave it blank, and I'll show you what that means in a tiny bit here. You're going to show all possible gametes of each parent, and then you're going to create a Punnett square. And you're going to show me the genotypic and phenotypic ratios. And so these are the steps you will follow. I'm going to walk you through a couple examples just to help you get the idea of what this should look like. So we're going to determine the cross of an F1 generation. So I'm going to cross a pure tall and a pure short together. I'm going to create my legend. My legend means big T is for tall because that's my dominant trait, and little t is short for my short trait. Now you will notice I'm not using two different letters here. Both letters are T. And the reason is because it's one trait. I'm talking about one thing. So I want to indicate to whoever's looking at my problem or whoever's watching or learning that I'm talking about the same thing, height. And I'm going to represent it with a T for tall and a little t for short. The uppercase and lowercase mean dominant and recessive. So there's my legend. I now need to write down the genotypes of my parents. Pure tall means that both of the alleles that the parent has have to be the same. So they're going to be big T, little t, big T, big T. The pure short means, again, they have to be the same. So there's two little t's. I'm now going to create what's called gametes. Essentially, that is taking what the genotypes of the parents are and splitting them out into what would be essentially an egg or a sperm. So each parent has two alleles. So there's two gametes from one parent and two gametes from the other parent. That's the first part of the setup. That is showing you the legend, who the parents are, and the gametes that they make. Making the gametes, guys, is very important because it allows you to set up your Punnett square. Punnett square is shown on the side here. What you're going to do is you're going to separate out the gametes and you are going to put one from one parent on the top and the others from the other parent on the side. Do not mix them together. Keep one parent in one area and one parent in the other. So you can see I chose to do the big T's on the top and the little T's on the side. But again, these were originally together and these were originally together. I'm now going to combine them into the boxes. And it's kind of like using a multiplication chart. This times this gives me this. This times this gives me this. This times this gives me this. And then that. So that is a Punnett square. And that is showing the setup of how I would do a monohybrid cross. A phenotypic ratio is now asking you to tell me what you see of all the plants. So when I look at this, I know all the plants are tall because they all have a dominant gene. So if I was to look at the plants, not knowing their genotype, I would see they are all tall. A genotype is what is the actual genetic setup of the alleles. So I notice they are all big T, little t. We call that heterozygous, meaning they have a mixture of both. So I would let you know that I have 4 heterozygous to no dominant tall to no dominant short. Or recessive short, sorry. So that is the setup every single time. Legend, genotypes, gametes, Punnett square, ratios. So we're going to do it one more time. But this time, reading very carefully, it says determine the F2 generation. So F2 generation means I have to take 2 plants that I just created in the F1 and I have to cross them. So I'm taking 2 individuals from the F1 generation. Both look tall, but we know that they were heterozygous genetically. So I'm going to create my legend again. Big T for tall, little t for short. Now, ladies and gentlemen, when you are creating legends, you can choose any letter you want, okay? As long as you show which one is dominant and which one is recessive. I always tend to lean towards using the dominant word's first letter. So T for tall. That's usually how I work it, but you can choose your letters. Just remember, if it's a dominant and recessive trait, you got to keep the letter the same. Just one's uppercase, one's lowercase. So now I'm going to write down the genotypes of the parents. I know the genotypes from the parents from the first cross were heterozygous, so big T's and little t's. I'm going to create my gametes. There's one set of gametes from parent 1 and one set of gametes from parent 2. I'm going to take the egg, put it up here. I'm going to take the sperm and put it down here. So again, keeping the eggs together and keeping the sperm together in the Punnett Square setup. I'm going to cross them. So this times this gives me 2 big T's. This times this gives me a big T and a little t. You will notice that I went this times this, but yet I wrote it as if I went this times this. Always put your big letter, your dominant trait first, unless you don't have a dominant trait. So again, this times this, and then 2 little t's. So now when I'm looking at my Punnett square, I can see I have 3 out of the 4 individuals that are tall, which means that I have 3 tall and 1 short. That is my phenotypic ratio, a 3-to-1 ratio. I can also see that if I look closely, I have a variety of genetic traits. So that's my genotypic ratio. I have 1 set that is a pure tall, so I have a 1. I have 2 that are heterozygous, so that's a 2. And then I have 1 that is a small one, which is small t. So I have a 1-to-2-to-1 genetic ratio or genotypic ratio. So what is a Punnett square? I've used them now. I've showed you them. They're essentially a diagram that analyzes the outcome of breeding experiments, and we are going to use them throughout this entire chapter. It helps us determine ratios and percentage outcomes. It's essentially a tool that tells us what are the possibilities of breeding 2 things together. Phenotypic and genotypic ratios allow us to see what they look like and what their genetics are. So we're going to try this out together and talk about how we can see what is the genotype and phenotype of each parent. So what you're seeing here are 2 short plants. If you look across the board, they're the short ones out of the process. We know they both have 2 little t's. If you look at it, you will see there's only one combination or one outcome that could potentially happen, and that is all their offspring are short, so they all have little t, little t. So that's A. B is showing 2 tall plants. Again, we know tall is a dominant trait. I have it over on the side there. So I have 2 tall parents. You'll notice I left a blank beside the big T. And that is because if I'm just looking at this plant, I'm just looking at them. I don't actually know if they're big T, or big T, little t. I can't, it's not screaming its genetics at me. It's not saying, I'm homozygous dominant, I'm heterozygous. It's just a tall plant. So I know it has the tall gene. I just don't know if it's heterozygous or not. So when I breed them together, I end up with all tall offspring. At this point, I can make an assumption that it's probably big T for both parents, but because I'm not 100% sure and there's not a lot of data there, I'm not going to guess. I'm going to breed now a tall and a short. So I know little t, and there we have it again, guys. I do not know if this is a homozygous dominant or a heterozygous individual. So I'm just going to not have the second letter there for the tall one. All in offspring end up being big T, little t. They're all tall, so I combine them together. My last set is a short and a tall. And now, ladies and gentlemen, I can tell you definitively that this one is a big T, little t, because if we look at their offspring, I have a short individual in that offspring. The only way I can get this to happen is if I have a little t from this parent combined with a little t from this parent, because the Punnett square sets it up to show you the crosses from these genetics going across. So I can say, yes, for sure, this has to be a heterozygous individual because we had a individual that is short with the recessive genes. There's a couple practice questions that are in your notes that we are going to try together very quickly. There's just a couple, and then I'm going to get you guys to do your own practice questions, and if you have questions, you can ask. So we're going to try the following monohybrid cross using a Punnett square. We got to find the phenotypic and genotypic ratios of that cross. So again, make your legend. Tell me the genotypes of the parents. Create your gametes. Put your gametes into your Punnett square. Cross it. And there you have the very first start. Now it says find the phenotypic and genotypic ratios. So I'm not done until I tell you the ratios. Looking at this, I can say the phenotypic ratio is too tall to too short. Why? Because I have two talls here because of the dominant trait and two shorts because of the recessive traits. Genotypically, I also have a 2 to 2 or 1 to 1 ratio because I have two that are the same and two that are the same. I'm always going to want to drop my ratios down to the lowest common denominator possible. So if it's 2 and 2, that can be reduced down to 1 and 1. So brachdactyly, is something called short-fingeredness, is actually a dominant trait after having normal length fingers. A brachdactylous man, he's heterozygous, marries a normal woman. What is the possible genotype and phenotype of their children? This is a typical example of a genetics question, in which you have to pull out information. So we need to go through step by step to find what we are looking for. So first thing first is I'm going to create a legend to help me out. Big B for brachdactyly and little b for normal finger. Now I need to figure out what are the gametes of my parents. So when I'm reading this, it says there is a man who is heterozygous. I would stop there and I would write down his genetics. So big B, little b. He marries a normal woman. Well, normal is recessive, so it would be little b, little b. So first I make out my genotypes of the parents, highlighting, underlining, figuring out what is step by step the parents' genotypes. I'm going to create my gametes, and then I'm going to put them in my Punnett square. I'm going to cross them, and I'm going to give you my phenotypic ratios. So again, I have 2 to 2 or a 1 to 1 ratio and a 2 to 2 or 1 to 1 ratio. So the possibilities of them having children is a 50% chance that they will be brachdactylic or 50% chance that they will be normal, is essentially what this is showing you. So Mendel kind of had some rules that he had to follow. So heredity is controlled by genes, and each parent has at least two different alleles for a trait. So essentially a big B and a little b, or a big T and a little t, or two little t's. So what he said is that the law of dominance is that the dominant allele will always mask the effect of the other allele. So if you have a dominant allele, that is what you are going to see. He also said that in organisms that are diploid, so their chromosomes have pairs like we do, okay, they will separate into different gametes during meiosis, which you know, you've covered meiosis. You understand that eventually you will have 4 sperm cells at the end, each with 23 in each sperm cell, or an egg of 23 chromosomes. And the last thing he's saying is the law of independent assortment is essentially that when you are going through the process of meiosis and you are creating those gametes, okay, or you're going through the process of mitosis and you're creating those cells, these lovely chromosomes don't have a rhyme or reason. They just line up randomly. Okay, so they line up randomly at the metaphase plate and then split into more cells, and it's all random. There's no set pattern to it. So that is essentially monohybrid crosses. There's a bunch of practice questions you now need to run through. If you have questions, please let me know, and we will talk to you soon.